again, I'm Matthew Lorence with Chimera Therapeutics, and our mission is to cure autoimmune disorders, which have no known cause, no established cures, therapies that only treat the symptoms, yet they impose a significant economic burden on the healthcare system. If we look at just 2 of more than 100 known autoimmune disorders, relapsing remitting multiple sclerosis and type 1 diabetes, these 2 alone impose a $4.4 trillion impact on the healthcare system in the United States with over 2.5 million patients. Mixed chimerism, which is where a patient's immune system and a healthy donor's immune system coexist in the patient, can cure autoimmune disorders. In practice, healthy donor stem cells are processed for transfusion into the patient, where they embed or engraft in the bone marrow, resetting the immune system and reestablishing immune tolerance. Damaged tissues and organs can now begin to regenerate and restore function, potentially curing the disease. Chimera Therapeutics' business model is to provide a fee-for-service processing of these donor stem cells, much like the CAR T companies, with a target price of $200,000 to $250,000 per treatment. Now, if we look at the competitors, especially in MS, they're the big pharma. You've probably seen their ads on TV, but these therapies only slow progression of MS. They do not cure the disease. With type 1 diabetes, again, the big pharmas supply synthetic insulin, which address the symptoms of the disease, but don't slow the progression of the underlying conditions that impact the patient's quality of life and lifespan. The Chimera Therapeutics team has 2 co-founders on the faculty of the City of Hope, including Art Riggs, one of the pioneers in the biotechnology industry, and world-renowned medical advisors who are experts in T-cell immunotherapeutics, immuno-oncology, and bone marrow transplantation. We're currently focused on a few autoimmune disorders, but the key is we believe this is a platform therapy because the same patient conditioning and treatment protocol will be used regardless of the indication. We're currently seeking a $10 to $15 million round. Funds will be used to match California Institute for Regenerative Medicine grants for phase 1 and phase 2 trials, and to establish our cell processing center in Southern California. In summary, mixed chimerism can cure autoimmune disorders by resetting the immune system and establishing immune tolerance, where damaged tissues and organs can begin to regenerate and restore function. In a phase 1 trial currently ongoing at City of Hope, we've demonstrated that the therapy is safe and non-toxic, and we've actually cured a sickle cell patient. All the IP for mixed chimerism has been exclusively licensed by the company from the City of Hope, and we're currently collaborating with them on 2 clinical trials. Thank you. Any questions?
A question for you. Are you considered a therapeutics company or a CDMO?
We think it's a therapeutics company, but we're obviously not making a pharmaceutical small molecule or a peptide to package. But
you're doing outsourcing for others as well.
Well, the idea is to do the service within the laboratory because we want to protect the IP, not just through the patents, but also through trade secrets. So it really is a service laboratory in that respect.
No, I was asking because there are very different valuation models for those. That's
okay.
Are the donors HLA matched? Like, how do you prevent graft versus?
So they're haploidentical donors, half matched, and the ability to establish the mixed chimerism in the patient actually prevents the onset of graft versus host disease, both chronic and acute.
Can you give us more details on the phase 1 trial? How many patients, some of the preliminary data?
I'm sorry, I didn't quite understand all of that. Oh. Excuse me.
Can you just give us more details on the phase
Sure.
1 trial?
So we had 3 patients in the phase 1 trial so far. It's been funded by CIRM for 6 patients. The first patient, the conditioning treatment was not strong enough. So while the graft took, it didn't last. The second patient, which is the one who was cured of sickle cell, they're now more than 2 years out. They show a stable mixed chimerism and they're free of symptoms from sickle cell. What I'd like to do is shift to this slide. So if you pay attention to anything on this slide, look at the green bars. Those green bars are naive T cells. So these are the cells that are not activated, and there's two critical factors here. This is indicative of an immune system that's in tolerance status. So critical for resolving any kind of autoimmune disorder. The other key thing is this is an indication of a young immune system. So while the majority of the cells in the patient are from the donor, who is his father, what we're seeing is an expansion of naive T cells, indicating that in some ways we've rejuvenated his immune system. The third patient, since you asked, mixed chimerism was stable. The patient was released from the hospital, but unfortunately died of an underlying condition. FDA investigated completely and cleared the therapy of anything with the resulting in that patient's death.
Can you talk about how that works for type 1 diabetes, where the islets of Langerhans are gone, basically?
So in preclinical models, what they've been able to demonstrate and work is that once the immune system has been reset and autoreactive T cells and antibodies are gone, beta cells in the pancreas, which appear to be almost senescent, are reactivated and start producing insulin. And some alpha cells in the animal models actually converted to beta cells and began producing insulin. So again, it's a mouse model, but we do think that there's good potential for this being duplicated in humans.
Why'd you choose aplastic anemia as your beachhead? And so
Thank you. Great question.
Okay.
So the concern is because this is like a bone marrow transplant, but the conditioning is much more mild than a standard bone marrow transplant. There's probably going to be a concern from the FDA about safety. So we wanted to target the first autoimmune disorder as something for which there was essentially no cure and relatively fatal. So we thought that would lower the bar for the FDA. Plus, it's both a hematological, although primarily an autoimmune disorder at the same time, small patient population, but the key thing is the PI who will be chairing the study at City of Hope has already identified prospective patients. So once the IND gets through FDA, which is being submitted this month, and we get the funding from CIRM, we'll hopefully be able to move into phase 1 this year. That's the plan. And did you ask about another disorder?
Well, I think I was going to ask after aplastic anemia, where do you
MS.
see this technology?
Yeah, assuming that we get the positive results we expect to see from the aplastic anemia trial, we immediately want to try and go towards an MS trial.
Can you speak to a little bit about reimbursement or how the insurance companies view
So
it?
that's also a very good question. So I don't know if you noticed, CMS just had a ruling on Chimera and Yescarta. Those are $373,000 and I think $475 that they're being charged. But there was an ICER study this summer where they essentially modeled the appropriate reimbursement based on the economic benefits of those two drugs, and they came out at $233,000. Kind of why we picked the $200,000 to $250,000 range for the reimbursement of our therapy.
Can you speak to the capital financing plan after the seed?
Sure. So the key thing is to set up the laboratory. That's what we need for the seed. We don't anticipate having to expand the laboratory dramatically because the processing of the cells is a relatively straightforward process. We think that we can manage several patient specimens per week. And these, because they're coming from the donor, the plan would be to cryopreserve and then ship the cells to the treatment center. Right now it's City of Hope. So we could see an expansion of the laboratory as volume increases, but we're not really anticipating much more at this point. By the way, it's commercially available equipment as well and reagents that we use.
I guess to the same line of thinking, in terms of your business plan, do you plan to do everything in-house, or are you thinking about maybe partnering with Resilience? Or
also, thank you. Good
platforms?
question. So initially, we want to keep everything in-house. Again, we have the IP, but we want to make improvements in the protocol, and there are some basically tricks within the protocol that are not clear from the IP. And so to protect it, we really want to keep one processing center in the United States. But obviously, there are countries like China where you can't import or export human tissue. So when we start moving into countries where there are those types of restrictions, we probably will set up a subsidiary facility to handle the processing in those laboratories. But within the United States, we hope that we can manage any partner treatment center directly from that one lab. Any other questions?
Maybe just one more quick
Please.
question. So I guess the other part of this is markers that you'd be tracking. So like, because in terms of diabetes, I'm thinking probably 2 or 4 antibodies for sure that you'd be tracking as the course of therapy goes forward in terms of if the levels of those antibodies go down. Is that the plan in terms of?
So what we're doing right now is we're tracking the CD4 plus and CD8 plus T cells in the patient.
Okay.
And what we're looking for is the presence of the autoreactive T cells and antibodies from the B cells in the patient. So right now, in terms of tracking insulin production, that's something that we would explore later when we get to that point. But I'm afraid type 1 diabetes, because the relative safety of treating with exogenous insulin versus a partial bone marrow transplant means that trial is going to probably be later until we establish how safe the therapy is with FDA.
True, but you'd have nice molecular markers like C-peptide to track. So
I'm sorry.
you would have nice molecular markers like C-peptide to track that.
We
cells
have
are-
molecular markers for the immune cells,
Right.
yes,
Yeah.
absolutely.
How are you measuring autoantibodies?
We're just so right now we're not measuring the autoantibodies themselves. We're just looking at the expansion of the immune cells in the patient. So the standard routine after the patient, the one that's been cured, was discharged from the hospital, comes back for blood draw every 6 months, and the level of the immune cells is checked in the patient. I think I probably used up a lot of time.
We got about 1 more minute. There's some more
Okay.
questions. Yeah.
Would you mind describing the process from the biopsy of the donor all the way to transfusion?
So it's mobilization of stem cells, which are collected by apheresis and then pooled. And then now you're in the secret sauce, which is, sorry.
Magic happens.
So yeah, that's when the cells are brought into the laboratory. So basically what happens is that we don't manipulate the cells. What we do is we remove a subset of immune cells and enrich for other subsets of immune cells that promote engraftment in the patient. And the cells that we remove are the ones that seem to promote graft versus host disease in the patient.
And that's all within a closed system, like a disposable system, or is it an open system?
It's done in and I'm sorry, I don't know the specifics of the system, but it is done in a GTP laboratory. All right, thank you very much.